Hi, my name is Phil Yules and I'm a bioinformatician working at the National Genomics Infrastructure in Stockholm, working with the Applications Development Group, and we develop new protocols and new bioinformatics analysis techniques which we bring into the main production side of our sequencing facility. I'm going to briefly go over our new RNA-seq analysis portfolio here, the best practice analysis that we offer to our users if you come in with an RNA-seq project. So RNA-seq is the most common type of sample that we process at NGI Stockholm. We have a lot of projects coming in every year, and there are also very numerous samples as well. So we process very many thousands of RNA-seq samples every year. So it's a large, very highly automated system, and it's pretty robust, and we're quite confident with it now. We have two protocols that we use in production, both produced by Illumina, one for poly-A sequencing and one to do ribosomal RNA depletion with a RiboZero kit. So TrueSeq poly-A is best for if you're interested in messenger RNA and you have high quality RNA, and RiboZero is great if you want to get all of the RNA fraction in a cell, or if you have degraded RNA. In the development group, we're also working on a few extra kits. There's the Clontech Smarter Pico, Illumina RNA Access and RiboZero kits, and these are good for low quality and low input samples. This is just a couple of slides from a recent development project where we've looked at replicate samples from test RNA and also very low quality FFPE samples with both the Clontech and the Illumina Access kits. On the left you can see the raw data clusters by the kit used, but then if we look at the counts over the genes themselves on the right, you can see that they cluster by biological samples, so the reproducibility is really high using these kits, despite the low quality of the RNA. If we look at one sample which has been prepared in replicate here, this sample has a RIN score of 2.5, a DV200 of 21%, so it's very low quality RNA, and yet we're able to pull very high reproducibility of gene counts between these two replicates. So if you're interested in this, please come and let us know and we can hopefully work with you on such projects. So however you generate your RNA samples, the NGI Stockholm offers a bioinformatics best practice analysis, kind of free of charge if you choose to have it. And we run this internally anyway to ensure the quality of your RNA samples. Those of you who have run RNA samples with us in the past will be familiar with our old pipeline, and it takes the raw FastQ sequencing data as inputs, and it gives you a range of results. So you have alignments in BAM formats, we have counts from genes, and we have a lot of quality control metrics. The first pipeline has been used in NGI Stockholm since 2012. It's one of our oldest best practice analyses, and basically is ready for an upgrade. We had three major focuses when we were doing this. We wanted to update the tools used and add extra functionality into the pipeline, and we also thought it was important to allow people outside of the sequencing facility to run the pipeline themselves. For instance, if you have samples of your own that you want to compare to. So taking a quick look at the tools and the functionality, the first pipeline ran through these different tools to take the data from its raw read states through to QC and alignment steps. The new pipeline does very similar steps, but you can see we've switched some of the tools that we use. Importantly, we've used switch top hat for star, which is commonly the most commonly used tool by many groups these days. It's much faster and gives comparable results. We also have switched from using cufflinks to string tie to give FPKM scores, which is developed by the same group but is more up to date. We have updated reporting and some updated QC steps. We've also added in a few extra steps into the pipeline, so we now do adapter trimming routinely for all samples. We do an extra QC step that looks at the levels of duplication in the reads and how this is related to the expression level of that gene. We've added in additional QC plots from our seq QC package. we've added in additional sample similarity tests to do with an MDS plot and a clustering plot. And all of this is bundled together in the end. This is an example plot from DupeRadar, the new tool which gives quality control metrics about the level of duplication in the reads. And along the x-axis, every point on these graphs is a gene, and the x-axis shows the level of expression for that gene, and the y-axis shows the percent duplication of the reads in that gene. The higher the expression of a gene, the more likely we are to see reads as duplicates. So on the left, you can see there's low duplicates for lowly expressed genes and high percent duplicates for highly expressed genes. On the right-hand side, we have a poorer sample, maybe a very low input or low quality, and you can see that the whole library has a pretty high level of duplication, irrespective of the expression level of each gene. So this is a new QC metric, which we hope will help identify problems in the library preparation steps of RNA samples. Another new analysis we've brought in is counting the types of genomic features that your reads overlap. So these are called biotypes typically, and you can see in this plot taken from a multi-QC report that most of the reads are overlapping protein coding transcripts, which is what we want to see. This can help highlight problems such as ribosomal RNA contamination or other kind of weird things going on with the library. Here we have one of the new heat maps showing a sample similarities between all the samples, with a single failed sample at the top, which has very low sample similarity. And all of these QC metrics and an MDS plot, which this is much like a PCA plot. It's a way to show a level of similarity between different samples and clustering. All of these metrics are bundled into a multi-QC report. This is a tool mentioned in the other talks, which has been developed at NGI.com and helps to visualize QC metrics across many samples in one place for a whole project. So this file will be delivered to you along with your data, and you can just view it in a regular web browser and explore the different QC metrics yourself. Okay, I mentioned that we wanted to allow others to run the pipeline as well as us. This is really important, as I say, if you have old data, for instance, generated with the old pipeline, which you'd like to compare to newly sequenced samples, or if you want to compare your data to public data sets from other papers, things like that. It's really important that you can reproduce the results that we provide in your own hands. We use a tool called Nextflow to run the new pipeline, and this is kind of a workflow manager which handles much of the back end for the pipeline. This allows easier maintenance for us. It gives much better reproducibility for every run, and it's also very easy to run. It's a single command. This hopefully will make it easier for research groups such as yourselves to run yourself. So if you're running on UpMax or UpNext, as we do, you can start off by installing Nextflow. You just grab it off a web. If you're running on Bianca or somewhere that doesn't have an internet connection, it is also available as an environment module. Then you simply run this command. Now, there's a few parts to this command. The important bit is at the start, where you can see the SciLifeLab NGI RNA-seq. That's the GitHub name of a pipeline. All of our pipelines are stored open source on the web, and specifying this will tell Nextflow to automatically download the pipeline code. We then specify the UpMax projects, the genome we'd like to run on, and where our raw FASTQ files are. The default setup in the pipeline is for UpMax, and it knows where all of the alignment references are and uses all of the centrally installed software packages. So hopefully it's very easy for you to run. If you don't have access to UpMax, the pipeline also runs on pretty much any other cluster. Nextflow supports all of the normal commonly used queue managers such as Grid Engine and LSF, as well as Slurm. We've tested it on the cluster in Gothenburg and worked with Jakob and NBIS to help install all the software that the pipeline needs using Bioconda. So that's entirely possible. We can also run the pipeline using integrated Docker support. So this is a very similar command again, and we still install Nextflow, but we just specify -profile Docker. And if you have Docker installed, that will download a Docker image which has all of the software dependencies. And this is best for reproducibility because it runs exactly the same versions of environment and software every run. And you can run this on your laptop if you have a small data set. Finally, if you have bigger data sets, maybe your laptop isn't powerful enough or would take too long to run. So we've also packaged a pipeline so you can run in the cloud using AWS. This is something we're working on at the moment to try and polish a bit more, but it has a lot of potential for the future. If you're interested, please let us know. Okay, so in conclusion, we've just released a new analysis pipeline for all RNA projects. It's got more up-to-date tools, it runs faster, gives better QC, and we hope you're happy with the results. It's in production for all RNA projects now running at NGI.com, so if you order a new RNA project with us, you should get results back from it. And if you'd like to, it should be pretty easy for you to run the same analysis on your own data. All of the code's available on GitHub. To find the code, go to github.com and search for scilifelab/NGI-RNA-seq. And if you have other projects which are not RNA-seq, we're developing a suite of similar pipelines which all work in the same way. For small RNA-seq, methylation or bisulfite sequencing, ChIP-seq, and also cancer analysis workflow mentioned earlier is also using Nextflow and runs in a similar way. All of these pipelines are open source and released under the MIT license, so you're free to modify them and build upon them in any way you'd like. Okay, with that, I'd like to acknowledge the people who have worked on this project and helped out, and everyone who's been involved in feedback and development of these pipelines. If you have any questions or you'd like to try out the pipelines yourself, please let us know and drop us an email at support@ngisweden.se.